Okay, so get ready for this, because today we're going deep into the world of DNA.
Deep, deep.
But we're not, like, going back to high school bio, right? We're talking about, like, going from the basics
Right.
all the way to the crazy stuff, like gene editing.
Yeah. And what I thought was really cool about the source material for this deep dive is it's not just like, what is DNA? It
Right.
actually gets into how scientists are manipulating it.
I know, it's like science fiction becoming reality.
Yeah.
So before we get ahead of ourselves, let's start with the fundamentals.
Okay.
What exactly is genetics, and why should we even care?
Well, genetics is the study of genes, heredity, and like variation in living organisms.
Okay.
And genetic variation is like the diversity of genes within a population.
Gotcha.
And genes are made up of DNA,
Okay.
and they contain the instructions for building and maintaining an organism.
So it's like the instruction manual for life.
Exactly.
Okay.
And they're passed down, these genes, from parents to offspring, influencing an organism's traits.
Wow, so it's not just about inheriting like your grandma's curly hair or something like that.
Oh.
It's like understanding the building blocks of who we are and how we function.
Yeah, absolutely.
Okay.
Now, when we talk about the complete set of DNA in an organism, we're talking about the genome.
Okay.
And it's all the genetic material in an organism.
Right.
And it's organized into chromosomes.
Okay.
It contains the genes that code for traits.
So if the genome is the entire library, then the genes are the individual books, right?
Yeah, exactly.
Okay. But what I think is crazy is that the size of that library can be so different between different organisms.
Yeah, yeah. And what's interesting is that the size of a genome doesn't necessarily correlate with the complexity of the organism.
What?
Yeah, some plants have much larger genomes than humans.
Really?
Yeah.
So strange. Okay, so we've got the library and the books, but what about the actual structure of DNA itself?
Right.
The double helix.
Yeah, the double helix. So it's this elegant, twisted ladder-like structure made up of two strands of nucleotides. Okay. And each nucleotide has a sugar phosphate backbone and one of four nitrogenous bases:
Okay.
adenine, thymine, cytosine, and guanine. So A, T, C, and G.
Oh, right, right.
And these bases always pair up in a very specific way.
Yeah, it's like A with T and C with G. It's
Exactly.
like a molecular dance or something.
That's a great analogy.
But why is that pairing so important?
Well, that base pairing is fundamental to how DNA replicates
Oh,
and how the genetic code is read.
okay.
So it ensures that the information is passed down accurately from one generation to the next.
So we've got the basics down, but now things start to get really interesting.
Okay.
You mentioned earlier that scientists are now able to manipulate this code,
Yeah.
like taking, I guess, combining genes from different organisms to create this thing called recombinant DNA.
Yeah, so recombinant DNA technology has opened up a world of possibilities.
It sounds kind of scary.
Well, it might sound like science fiction, but it's a reality that's already having a huge impact. So think about GMOs, genetically modified organisms.
Yeah.
We're already eating crops that have been engineered for pest resistance or enhanced nutrition.
Oh, wow.
So that's just one example. But there are many other applications.
So GMOs are just one example.
Right.
So what are some other ways that we're using this recombinant DNA technology?
Well, there's gene therapy, which holds a lot of promise for treating genetic diseases by replacing faulty genes with healthy ones.
Wow.
And there's DNA fingerprinting, which is used in forensics to identify individuals and solve crimes.
It's crazy how much is possible.
I know, it's mind-blowing. Like, we're really on the cusp of some major breakthroughs.
So I have a question.
Sure.
How do scientists actually go about doing that, like stitching these DNA fragments together from different sources?
Right.
Is there like a molecular sewing machine or something?
It's not quite a sewing machine, but we do have this amazing enzyme called DNA ligase.
Okay.
And think of it as a molecular glue that joins DNA fragments together.
So it's like this tiny craftsman just piecing together these building blocks of life?
Yeah, it's a great analogy. And DNA ligase is really crucial for a lot more than just recombinant DNA technology.
Oh, really?
Yeah, it's essential for natural DNA replication and repair,
Okay.
keeping our DNA intact and functional.
Wow. So we've talked about some of the applications.
Right.
But what are some of the like key techniques that scientists use to study and manipulate DNA?
Well, there are a bunch of really cool technologies that are highlighted in your source material,
Okay.
like molecular cloning, which allows scientists to isolate and make copies of specific DNA sequences.
So we can zero in on like the specific genes that we're interested in?
Exactly.
Cool. What else?
And then there's polymerase chain reaction, which you'll often see abbreviated as PCR,
Okay.
and it amplifies DNA sequences exponentially.
Wait, so you're saying it's like you take a page from the instruction manual and then you make billions of copies?
Yes, exactly.
That's
It
insane.
is. I mean, PCR has completely revolutionized everything from medical diagnostics to forensic science.
Wow.
So, like, imagine you have a tiny DNA sample from a crime scene, right?
Okay.
Well, with PCR, you can amplify that to get enough material for analysis.
Wow.
And it can also be used to detect viruses and genetic mutations.
Wow, that is a game changer.
Yeah, it is.
So we've got the techniques for copying and amplifying DNA, but what about actually analyzing it?
Right.
Like, how do we actually decode the instructions?
Well, for that, we use techniques like gel electrophoresis and DNA sequencing.
Okay.
So gel electrophoresis separates DNA fragments by size, kind of
Sure.
like sorting books by height, and DNA sequencing determines the exact order of those A, T, C, and G nucleotides within a DNA molecule.
Oh, so it's like reading the instruction manual letter by letter.
Precisely.
That's pretty powerful.
It is really powerful. DNA sequencing has completely revolutionized genomics research.
Wow.
So, like, we can now decode entire genomes and understand the genetic basis of disease and personalize medicine based on someone's unique genetic makeup.
This is incredible.
I know, it is incredible.
And there's one more technique that you mentioned earlier that I really want to hear more about.
Okay.
CRISPR-Cas9 gene editing.
Ah,
This is like straight out of a movie.
it does sound like it's straight out of a futuristic movie.
Yeah.
But CRISPR-Cas9 is like having a molecular scalpel that allows scientists to precisely target and modify specific DNA sequences.
So wait a second. We're talking about actually editing the instruction manual of life.
We are, which is why it's so important to use this technology wisely.
Yeah, I can only imagine the ethical questions that something like that would raise.
For sure, for sure. So one of the things we have to think about are off-target effects, unintended changes to the genome that could have really unpredictable consequences.
So like those molecular scissors could accidentally make cuts in the wrong places?
Yeah, precisely. But scientists are working hard to improve the precision and safety of CRISPR-Cas9.
It sounds like this is going to be a really interesting conversation.
It is.
This is a lot to process, you
Yeah.
know? Before we dive deeper, I think it's important to make sure that we're all on the same page with some of these concepts.
Okay, sounds good.
So can we break down molecular cloning and PCR a little bit more?
Yeah,
I
absolutely.
think those are pretty key to understanding the rest of this stuff.
Okay, so let's start with molecular cloning.
Okay.
So you have a specific gene you're interested in, right?
Okay.
So you isolate that gene from the rest of the DNA using special enzymes called restriction enzymes.
So it's like molecular scissors.
Exactly, molecular scissors. And these restriction enzymes create compatible ends on the DNA fragment and on a cloning vector, which is a DNA molecule that can carry and replicate the inserted DNA.
So the cloning vector is like a little vehicle for transporting the gene?
It is. It's a great analogy.
Okay.
So the DNA fragment is inserted into the vector using DNA ligase, our molecular glue, and this recombinant vector is introduced into a host organism, like bacteria, which replicates the vector and the desired gene along with it.
So the bacteria becomes like a little factory for producing copies of the gene?
Exactly. And by isolating colonies that contain the vector and the inserted DNA, scientists can obtain large quantities of the cloned DNA for further study and manipulation.
That is so cool.
Yeah, it is.
Okay, so that's molecular cloning in a nutshell.
Right.
Now let's turn to PCR.
Right.
So we know it amplifies DNA, but how does that actually work?
Okay, so PCR is like a molecular copy machine that works in cycles.
Okay.
So first the DNA is heated to separate the two strands of the double helix.
It's like we're unzipping the DNA molecule.
Exactly. That's a great way to put it. Then short DNA sequences called primers bind to specific regions flanking the DNA segment that you want to amplify.
Okay.
And the temperature is lowered and DNA polymerase comes into play.
Isn't DNA polymerase also involved in natural DNA replication?
Yeah, it's the same enzyme that copies DNA in our cells.
Wow.
And it uses those primers as starting points and adds nucleotides to build new DNA strands that are complementary to the original template.
Oh, wow.
And with each cycle, you're doubling the amount of DNA.
So it's like a chain reaction of DNA copying?
Exactly. So you end up with billions of copies, enough for all sorts of analysis.
And this amplified DNA can then be used for a ton of different applications.
Oh yeah, tons of applications. PCR has revolutionized everything from analyzing ancient DNA to diagnosing diseases to identifying individuals and forensic investigations.
Okay, so molecular cloning and PCR checked.
Yep.
Next up, I think we need to delve into gel electrophoresis and DNA sequencing, and then, of course, the mind-blowing CRISPR-Cas9.
All right, let's do it.
But for now, I think that's plenty for our listeners to digest.
I agree.
Stay tuned for part 2, where we'll uncover even more secrets of DNA.
I can't wait.
All right, so welcome back. Last time we talked about getting into DNA sequencing and CRISPR-Cas9.
Yeah.
So let's dive into DNA sequencing.
Okay.
We know that it determines, like, the precise order of nucleotides in a DNA molecule,
Right.
which sounds incredibly complex. So how does that even work?
Well, it is a complex process, but brilliant minds have figured out some ingenious ways to do it.
Okay.
Two common approaches that you'll see are Sanger sequencing and next-generation sequencing,
Sanger sequencing and next-generation sequencing.
or NGS.
NGS, okay. Those names sound pretty intimidating.
Yeah, they can be a mouthful.
Can you break those down for us?
Yeah, absolutely. So Sanger sequencing is also known as chain termination sequencing,
Okay.
and it uses special nucleotides called dideoxynucleotides.
Deoxynucleotide.
Yeah, that's kind of a mouthful.
Yeah.
And these dideoxynucleotides act like stop signs during DNA replication.
Stop signs.
Yeah, so they halt the process whenever they're incorporated into a growing DNA strand.
So it's like throwing a wrench in the DNA replication machinery.
Yeah, that's a great analogy.
But how does that help us figure out the sequence?
Well, by using a mix of normal nucleotides and these chain-terminating dideoxynucleotides, you end up with DNA fragments of different lengths,
Okay.
each ending with a specific dideoxynucleotide.
Oh.
So you can separate these fragments by size. And by looking at the order of those terminating nucleotides, you can work backwards to deduce the original DNA sequence.
So it's like solving a puzzle, looking at the pieces in a specific order.
Exactly.
Okay. And now what about NGS? How is that different from Sanger sequencing?
So NGS takes DNA sequencing to a whole new level.
Okay.
It's a high-throughput approach that can sequence millions or even billions of DNA fragments simultaneously.
Billions.
Billions.
That's a huge leap forward.
It is, yeah.
How does it even handle that much data?
Well, it uses massively parallel sequencing technologies, which allow for a massive increase in the speed and efficiency of DNA sequencing.
So we've gone from deciphering single genes to unraveling entire genomes.
Yeah, pretty much. And it's now possible to sequence entire genomes in a fraction of the time and cost that it used to take.
Wow, that's mind-blowing. What kind of impact has that had on research in medicine?
Oh, huge impact. So DNA sequencing has allowed us to understand the genetic basis of countless diseases, identify potential targets for new drugs, and even start to develop personalized medicine tailored to an individual's unique genetic makeup.
Personalized medicine based on your DNA.
Yeah.
It's like something out of science fiction.
It is incredible. And speaking of groundbreaking advancements,
Yeah.
let's move on to CRISPR-Cas9 gene editing.
Okay.
This technology has the potential to rewrite the very code of life.
Yeah, CRISPR has been making a lot of headlines lately.
Yeah.
You mentioned before that it's like a molecular scalpel.
Right.
Can you explain exactly how that works?
Yeah, so CRISPR-Cas9 is actually a system that bacteria use to defend against viruses.
Oh, really?
Yeah, so scientists have harnessed this system to create this really powerful gene editing tool.
Wow.
And it has two key components: the Cas9 enzyme, which acts like molecular scissors, and a guide RNA molecule that tells the Cas9 exactly where to cut the DNA.
So the guide RNA is like a GPS system for the Cas9 scissors. Like, it leads them to the exact spot in the DNA that needs to be edited.
Yeah, that's a great way to think about it. So the guide RNA binds to a complementary target sequence in the DNA, and then the Cas9 makes a cut at that specific location.
So the DNA gets cut.
Right.
Then what happens?
So that's where the real magic happens. Scientists can use the cell's natural repair mechanisms to introduce very specific changes to the DNA sequence.
Wow.
So they can insert new genes, delete existing ones, or even just make single-letter changes to the genetic code, and all with incredible precision.
So it's like we're actually editing the instruction manual of life.
We are.
So what are some of the ways that we could actually use this technology?
Well, the potential applications are vast.
Okay.
So in medicine, CRISPR could be used to treat genetic diseases by correcting faulty genes.
Huh.
Like, imagine being able to cure inherited disorders like cystic fibrosis or sickle cell anemia.
Yeah, that would be life-changing for so many people.
Absolutely. And CRISPR could also be used to develop models for studying diseases,
Okay.
allowing researchers to better understand those underlying mechanisms and to test potential therapies.
Oh, wow.
And in agriculture, it could be used to enhance crop yields, create pest-resistant varieties, and even develop crops with a better nutritional value.
It sounds like this technology is truly revolutionary.
It is.
But with such power comes a lot of responsibility, right? I mean, there must be some serious ethical questions about manipulating genes in this way.
You're absolutely right. So one of the concerns is the possibility of what we call off-target effects,
Okay.
which are unintended edits to the genome that could have unpredictable and potentially harmful consequences.
So those molecular scissors could accidentally make cuts in other places.
Yeah, it's a risk that scientists are working hard to minimize.
Okay.
They're trying to improve the precision and safety of CRISPR.
It seems like it would be pretty hard to control.
It is. It's a very complex technology, but it has incredible potential.
Yeah. Well, this is all fascinating, but my brain is starting to overload a little bit.
I can imagine.
Before we move on, I think it's important to touch back on some of these key points.
Okay.
Let's start with a really basic question.
Okay.
What exactly is a gene, and how does that factor into inheritance?
Okay, so a gene is essentially a unit of heredity.
Okay.
It's a segment of DNA that contains the instructions for building a specific protein or a functional RNA molecule.
Okay.
And you can think of it as a blueprint for a particular trait.
All right.
And these genes are passed down from parents to offspring, carrying information that shapes an organism's traits.
So genes are like the individual chapters in the instruction manual,
Right.
and then inheritance is like how those chapters are passed down through generations.
Exactly. And understanding inheritance can really help us grasp concepts like genetic variation,
Okay.
which is the diversity of genes within a population.
Yeah. Okay.
And this variation is what makes each of us unique and contributes to the diversity of life on Earth.
It's like having a peek into your own personal instruction manual, seeing what makes you tick.
Yeah, exactly.
Okay, so let's shift gears a little bit.
Okay.
Let's talk about PCR again. We've talked about its use in medicine and forensics, but I think it's just so important to emphasize how transformative this technique has been.
Absolutely. I mean, it has revolutionized countless fields.
Yeah.
So in medicine, for example, PCR is used to diagnose infectious diseases
Okay.
by detecting the presence of viruses or bacteria in patient samples.
Right.
It can also be used to screen for genetic mutations associated with diseases like cancer.
It's like having a microscopic detective on the case.
That's a great way to put it.
Okay, and then in forensic science, PCR is essential for DNA fingerprinting
Right.
by amplifying those tiny amounts of DNA found at crime scenes.
Right.
Investigators can create these genetic profiles that can be used to identify suspects, victims, even missing persons.
Absolutely. It's incredible how PCR can help bring justice and closure.
Yeah, it really is. Okay, so for our final discussion point, I think we need to go back to CRISPR-Cas9,
Okay.
gene editing.
Right.
It's just such a powerful technology, so full of promise, but also, like we said, some serious ethical considerations.
For sure.
So we've talked about the risk of off-target effects, the potential for unintended consequences.
Yeah.
But what are some other ethical concerns that we should be thinking about?
Well, one major concern is the potential for what we call germline editing,
Okay.
which involves making changes to the DNA that would be passed down to future generations.
Right.
This raises some pretty profound questions about our role in shaping human evolution.
It's almost like playing with the very fabric of humanity.
It really is, and we need to proceed very cautiously and have some really open and honest discussions about the implications of this technology.
I think that's a great place to stop for today.
Yeah.
This deep dive into DNA has been really eye-opening, going from the basic structure of this molecule to exploring its manipulation with all this cutting-edge technology.
Yeah, it's been quite a journey.
We've seen how DNA shapes life, how scientists are deciphering all its secrets, and how we're actually starting to harness its power in these amazing ways.
It's really incredible to think about.
So as we wrap up this deep dive, I want to leave our listeners with one final thought to ponder.
Yeah.
Imagine a future where gene editing is commonplace.
Okay.
Like a future where we can cure inherited diseases, enhance human capabilities, and even influence the traits of our children.
It's both exciting and a bit daunting to think about.
It is.
So what kind of ethical boundaries should we establish? What choices will we face? And how will those choices shape the very essence of what it means to be human?
It's a future full of promise and, like you said, potential pitfalls.
Yeah, for sure. And we have a responsibility to use our knowledge wisely and engage in a thoughtful dialogue about the future we're creating.
Thank you so much for joining us on this incredible journey into the world of DNA.
It's been my pleasure.
I think we've all gained a new appreciation for the power and complexity of this molecule and the challenges and opportunities that lie ahead. All right, so welcome back to our final part of our DNA deep dive.
It's been quite a journey, hasn't it?
It really has.
Yeah.
From the basic structure of DNA all the way to like mind-blowing technology, CRISPR. It's
Yeah.
just incredible.
Yeah, it's pretty amazing how far we've come, from those early discoveries to like these incredible advances that are happening right now.
It is, before we wrap things up, though, I think it's really crucial to circle back to those discussion points we've been talking about
Right.
because they really highlight why understanding DNA matters, like not just for scientists, but for all of us.
Absolutely. I mean, these are not just like abstract concepts. They have real implications for our lives, the choices we make, the future we're shaping.
Yeah, exactly. So let's start with that fundamental question.
Oh.
What is a gene and why should we care about inheritance?
Okay, so at its simplest, a gene is a unit of heredity.
Okay.
It's a segment of DNA that carries the instructions for building a specific protein or a functional RNA molecule.
Okay.
And these proteins and RNA molecules are the workhorses of our cells,
Okay.
carrying out all sorts of tasks to make life possible.
So genes are like the blueprints.
Yeah.
For the building blocks of life.
Yeah.
And then inheritance is how those blueprints get passed down.
Exactly.
Okay, cool.
So understanding inheritance helps us grasp concepts like genetic variation,
Okay.
which is the diversity of genes within a population.
Okay.
So this variation is what makes each of us unique, and it contributes to the incredible diversity of life on Earth.
That's so cool.
Yeah, it's pretty amazing, and it helps us understand why some individuals might be more susceptible to certain diseases than others.
So it's like having a peek into your own personal instruction manual,
Yeah.
seeing what makes you tick and what potential health challenges you might face.
Yeah, that's a great way to put it. And this knowledge can really empower us to make informed decisions about our health, family planning, even just like understanding human history and evolution.
Absolutely. Okay, well, let's shift gears a little bit and talk about PCR.
Huh?
We've already talked about its use in medicine and forensics, but, it's worth emphasizing just how transformative this technique has been.
Oh, yeah, it really has revolutionized so many fields.
Yeah.
So, in medicine, for example, PCR is used to diagnose infectious diseases
Right.
by detecting the presence of viruses or bacteria in patient samples.
Oh, okay.
And it can be used to screen for genetic mutations associated with diseases like cancer.
So it's like a molecular detective.
Right.
Sniffing out the clues that were previously hidden.
Exactly. And in forensic science, PCR is crucial for DNA fingerprinting.
Right.
So by amplifying those tiny amounts of DNA found at crime scenes, investigators can create these unique genetic profiles
Right.
that can be used to identify suspects, victims, or even missing persons.
It's amazing how PCR can really help bring justice and closure.
It really is, yeah.
Okay, so for our final discussion point, I think we need to turn to CRISPR-Cas9 again.
Right.
Gene editing.
Right.
Such a powerful technology full of promise, but also, like we said, a lot of ethical consideration.
Yeah, definitely.
We talked about the risk of off-target effects.
Right.
The potential for these unintended consequences. But, what are some other ethical concerns that we should be thinking about?
Well, one major concern is the potential for germline editing,
Okay.
which involves making changes to the DNA that would be passed down to future generations.
Right, that was the big one that we talked about before.
Yeah, and this raises profound questions about our role in shaping human evolution.
It's almost like playing with the very fabric of humanity.
It really is.
Yeah.
And so we need to proceed with extreme caution and have those open and honest discussions about the implications of this technology.
Yeah, for sure. Well, this has been an amazing deep dive, hasn't it?
It really has.
We went from understanding the basic structure of this molecule to exploring its manipulation with this cutting-edge technology.
It's really been incredible to see how far we've come in our understanding.
Yeah, we've seen how DNA shapes life, how scientists are deciphering its secrets, and how we're starting to harness its power in these, incredible ways.
Yeah, it's exciting and a little bit daunting to think about what the future holds.
It is, it is. Well, thank you so much for joining us on this incredible journey into the world of DNA.
It's been my pleasure.
I think we've all gained a new appreciation for the power and complexity of this molecule and the challenges and opportunities that lie ahead.